Welcome to the External Medicine Podcast. My name is Daniel Belkin, and this is my co-host and brother, Mitch Belkin. We are here with Saloni Dattani, who is a PhD student at King's College London and a founding editor at the online magazine Works in Progress. She is also an editor at Stripe Press and a researcher at Our World in Data. Welcome to the External Medicine Podcast, Saloni.
Thank you. Thank you for having me on.
Before we get started, do you have any financial disclosures?
So I work for Works in Progress at Stripe, payments company, and I also work at Our World in Data. Those are the two financial disclosures I think would be relevant here.
So you're doing your PhD at King's College in psychiatric genetics, is that right?
Yeah, so it's actually a joint degree at King's College London and the University of Hong Kong. So I grew up in Hong Kong and my parents still live there. And I thought, like, there's this opportunity to be at both places. I like being in London, but I also wanted to spend some time with my family. And so part of my studies was based there and part of it is here.
What is your PhD about?
So it started off as one thing and now it's a kind of different thing. So it started off being about, so I don't know if you've heard about the missing heritability problem. So essentially, heritability is something that can be measured in different ways. And what it broadly means is the amount of differences between people that can be attributed to differences in their genetics. So usually it's expressed as a percentage. So you might say that 80% of the differences in people's height is determined by differences in their genes. And that is usually measured from twin studies. So in twin studies, you can compare identical twins and fraternal twins. And because identical twins share approximately 100% of their DNA and fraternal twins share half of that, you can then look at how much the identical twins have similar heights versus how much the fraternal twins have similar heights. And then based on that, you know that, well, these two different types of twins have 50% of their DNA different. And so you can kind of do a regression to figure out how much of the differences in those two different types of twins is due to their genetics. So if the identical twins are much more similar than the fraternal twins are, then that implies that something is much more influenced by genes. And so this is the way that we traditionally estimate heritability. But there's also other ways to do it, especially because people are because of the cost of sequencing or genotyping people's DNA, you can often look at particular genetic mutations that they have and try to link them to their height, for example. But the problem with doing that was that we weren't really finding the same amount of variance. So with the twin studies, we found that the twin studies tell us about the total amount of genetic differences that are linked to their differences in height. Whereas when we're looking at their genes, we're not actually looking at every single base pair in their DNA. We're only looking at a small fraction of them just because that's cheaper to do. And also we're looking at a much broader range of people. So instead of twins, it's usually people who are unrelated to each other. And so you don't find the distinction between identical twins and fraternal twins that you would there. And the differences between unrelated people are much smaller, I guess, like the kind of contrast that they have. And what that means is that you tend to need very large sample sizes in order to find the variants that are linked to their height. So the sample size issue was one reason. Another reason was that when you're looking at DNA, you're usually only looking at the common mutations that people have rather than the rare ones. So common mutations are anything that are kind of defined as anything that at least 1% of the population has. And so you'd think, well, maybe the rare ones don't really contribute that much because they're rare. But in fact, although each rare variant is rare, most people have a lot of rare variants. So having any rare variant is quite common. And also, rare variants tend to have larger effect sizes than common variants. And the reason for that is the type of natural selection that goes into making variants common or rare. So if a variant has a really large and like it really changes your height in some way, that might be a huge disadvantage. And over many generations, there would be pressure for that variant to be selected out of the population. And so the variants that remain that have very large effect sizes are the ones that have now become quite rare. So my PhD started off looking at this problem and trying to figure out how much of the heritability in psychiatric traits was not being found by these genome-wide association studies because we weren't looking at the rare variants. But in the end, I realized that I kind of just like the meta aspect of it much more, of just depending on how you designed your study, how many people were in your sample, or which specific things you're measuring, you could find different results. So you might just miss lots of variants that were relevant, or maybe you have people in your sample that other people didn't have, or like are selected in some way where you're only looking at healthy people or something like that. And that, how does that change the results that you might get? So my PhD eventually turned into this kind of meta level study. And so I'm kind of have been looking at different types of measurement, different types of samples, and how all of that affects research in the genetics of psychiatry.
So you brought up a lot of really, really interesting topics there. So when you're actually looking at these genome-wide association studies, how much of the genetic sequence are actually extracted from that? I'm sort of curious. I've never really thought about that in any detail, how much information we're leaving behind versus how much we're retaining to look at the single nucleotide polymorphisms.
Right. Well, in so there's two types of kind of broad types of ways, or three, that we look at DNA. So the first is sequencing, where you try to get every single base pair in someone's DNA. In reality, we haven't actually been able to do that until very recently, because the telomeres and also the centromeres of chromosomes are really difficult to sequence. And the reason for that is they have like these regions that are very repetitive. And when we're sequencing, we're only looking at little fragments and trying to put them together to find out what the entire length of somebody's genome is. And so when it's very repetitive, you don't really know where those repetitive sequences go, which order they're in, or anything like that. And so until this year, we weren't actually able to sequence the entire human genome. But that's one way to study it. And then the other way is looking at exomes. So exome sequencing just looks at the exons of genes, which are the bits that are expressed. And so there you would be looking at most protein coding genes, and even within those, only the exons. But even then, so it would be every single base pair of each exon in genes. And then genotyping is a much cheaper and like lower coverage way to look at people's DNA. And that is usually, I think it's usually a few 100,000 or a million or so base pairs, where these are mostly the common variants that people have that distinguish them. But also, you're not looking at every single base pair. You're looking at bases that have quite large gaps between them. And And then what you often do is you use those the information of those variants that are spread apart to kind of impute the missing variants in between. And to do that, you have like a reference panel where you have sequenced their entire genome, and you compare the bits that you have from your sample to the people where you have their entire genome, and then you try to compare, like if you find matches in the variants that you've looked at, you just then fill in the gaps to varying degrees of accuracy.
Interesting. So this is a podcast about interesting or big ideas in medicine, and I'm guessing most of our audience has not really thought about the genetics of psychiatric disorders very much. What are some interesting things that, as an expert in that field, you could share with us, teach us and our audience?
Sure. I would say like there are things that are common between lots of psychiatric disorders in terms of their genetics, and then there are things that are quite different. So the thing that's common is that with many psychiatric disorders, taking autism and schizophrenia as examples, with them, there's often two different like pathways in which you might have those conditions. One is you might have particular mutations that have very large effects, and those tend to be like very large structural changes in your chromosomes. So having entire segments that are duplicated or deleted during replication, and that might lead to developing the condition. And then the other pathway is to have lots of genetic variants that are related to that condition that each have small effects. And so, I mean, you could also have the combination of both and they add up. But I think what people often think is that it's just like one gene that causes autism or something, and that's not true. But it's also a bit more complicated because sometimes specific genes can have very, very large effects. It's just that that's not often the way that people would have that condition. They would often have many, many variants with very small effects each.
I'm curious how, when looking at something like, say, schizophrenia or autism, which are considered to be diagnoses, but are extremely broad, both in their presentation and features, how one would go about classifying things. Because if you're looking at, say, genetic studies to try to understand the underlying condition, we're assuming that this is one thing rather than multiple different but somewhat similar things.
Yeah, definitely. So I think the thing to remember is that what you're defining as like a case of autism or schizophrenia then determines how well you can predict it. So if you have a very broad, like if you're just looking at autism spectrum, which could be, like severe autism, or it could be like people who are able to go to school and have a normal job. If you're then trying to predict that in a new sample, then you're also going to be predicting those same kinds of that whole range of traits. And so going into more specific subtypes would then lead you to find a smaller range of genes that are linked to it. But also, if you look at, so I think there have been a few studies where people have tried to simulate how misclassifying or like misdiagnosing people would affect the results of a study. And so with schizophrenia and bipolar disorder, for example, they have some shared symptoms and features. And so if you then have like a high rate of misclassification between them, you also find different genes that are linked to them. You find like a mix of genes that are like linked to both bipolar disorder and schizophrenia. compared to samples where they're looking at only one of these conditions and excluding any symptoms that are related to the other very well.
So that would kind of hint at evidence that I think has been accumulating over the last few years of some architecture for mental health, genetic architecture for mental health, where genes that are associated with a bunch of different disorders, even ones that you wouldn't necessarily expect, end up being present in many of them.
Right. Yeah. So there's definitely lots of similarity. There's also some that is like similarity just because of the artifact of misclassifying people. I think the other thing that's important to remember in terms of the genetics of psychiatry is that people often think that if they have a genetic risk for developing a disorder, then it's kind of inevitable that they will develop it, or that doesn't like change over time, or it's something that's kind of fixed or determined in some way. And that's often not the case. So with like giving some very basic examples, with height, for example, height is highly heritable. So 80% of the differences between people's height is determined by differences in their genes. But at the same time, human height has increased quite a lot, even in the last century. And you can't really explain that by saying people's genetics have changed massively. It's actually because of them having better access to nutrition, better healthcare, and so on. And so the way that I tend to think about that is that heritability is telling you about the differences between people within a population, but also there are things that can shift the entire population on a trait. So everyone could have more nutritional access, and therefore everyone's height increases. And so the differences between them might still be highly influenced by their genes, but you can still make a big impact with these environmental factors. And then that's also true with medication. So if there's access to like a new drug for heart disease or something, then even if you had a predisposition to it, you may not develop the condition if you were using this drug, for example.
Let's turn over and talk a little bit about a topic that you have thought a lot about, which is improving science. What are some of the areas in science, be it funding, research, publishing, replicating results, regulation? What are some of those areas that you think are the most dysfunctional currently?
Yeah, that's a really good question. I feel like in comparison to other people who talk about these problems, I'm relatively optimistic about how science currently is functioning. But at the same time, I think there are a lot of things that I think just don't work very well, not intentionally, but just because of some kind of like pathway dependency. This just happened to be the way that things turned out. One thing that I think is a big problem is that there's often a lack of division of labor in science. So often people are expected to do everything. So if you're if you're a PhD student, you're expected to read and understand the literature, understand the fundamental theories and all of the background knowledge in your field. You're expected to know how to code, manage your data, give presentations, organize, like apply for grants, do all of these different things that require quite a lot of time to get used to. There's a very steep learning curve for programming, at least, I think. But you're expected to do that quite early on and manage to put together big research projects when you're just starting out. And I think that makes everything quite hard. And I think it means that people who recognize that they're good at particular aspects aren't able to then focus on that. So you have this expectation to keep up with the latest developments in every aspect of these skills that you're expected to learn. And there's often not that much collaboration that happens between people of different skills. You often have collaboration between people who are working on similar projects in the same field, but not like you wouldn't often work with someone who was doing data management as their main career, or you wouldn't be working with a software engineer, or you wouldn't often be working with a clinician, for example. And all of these things take a lot of experience and time to learn. And I just think, having teams where you have your own expertise and you're working with other people who have their expertise and working together on a project is much more efficient, and also would improve the quality of a lot of scientific publishing.
Interesting. Do you have any thoughts on how to improve that?
I think it's like I feel like the reasons for that are things that make it kind of difficult to change. So I think one reason is that when we have scientific labs, people that you're often recruiting to join your labs are not people without academic qualifications. So, for example, at our role in data, I work with people who are software engineers or who work with data management pipelines all day, and that it's really great to be able to have that. But that's not the case in academia, where almost everyone in a lab has a PhD background or has gone through the same kind of training in their undergrad and graduate school. So I feel like one way is just to open up the field, kind of recruit people who have completely different backgrounds, but who have the skills that you need and don't have. And then another aspect is often the pay and the flexibility that is a part of academia versus working in other fields. So I think often with people who have the background in programming, they often have much better career options in terms of salaries than they would in academia, or better perks and flexibility, and so it just wouldn't seem like an option for them.
It's interesting to think about the division of labor. There not being enough division of labor when it comes to science. I feel like I've also heard the perspective that's almost like saying that people are too fox-like and we need more hedgehogs within the field. And it's funny that I hear from the people who are most talking about foxes and hedgehogs, there seems to be a celebration of the fox more so, that people can kind of get siloed and study their one particular thing way too far, and they don't see the relevance for fields outside of it. It's kind of it's interesting to see, these are also downsides of being too fox-like in a way.
Right. Yeah, I guess the way that I think about it is what's more difficult is to develop these skills and tools rather than the knowledge or how you can apply that to different areas. So often I think it works better to have a team of people with someone who's a researcher, someone who's a writer, someone who's a data engineer or programmer, but all of them are working on different projects that then might be in completely different fields. And that I think is much easier to do because the amount of learning curve that you have with the skills, especially programming, for example, it just takes much longer to develop that across a team than you would to learn about a new field, which you can kind of get by going to lectures and talking to people who are involved.
Besides the division of labor issue, are there any other aspects of science that you think are dysfunctional or have put some thought towards how to improve upon?
Yeah, so the other big one that I've written about is peer review. So I think often when people talk about this, they kind of make it sound like peer review is bad. But I actually think peer review is really good and really important and fundamental to how we do science. And what I think is that it's not rewarded enough and not in the right ways. And so what currently happens in scientific publishing is that when you have, you've done a big project and you've written that up as a paper, you'll send it to a journal that you think would be the right fit for it. And the editor will then send it off to a few other scientists in your field that they think would be experts or would be able to review the paper. And all of those other scientists have full-time jobs as well. They have their own research projects that they're working on. Often they're doing teaching and managing a lab at the same time. And so you often don't know how much time they have to spend on reviewing your work, whether they have any time at all. And the editors will kind of wait for weeks maybe to get a reply from them on whether they're interested in reviewing, still try to find other reviewers and so on. And so there's this time management issue that means that once they actually agree to review or provide comments on it, it might be several months before you hear any response back from the editors. But the other thing is that you often don't even know about the quality and the interests of the reviewers that you've contacted. So, someone who's in peer review could be a PhD student, it could be a senior professor. Often journals will ask you to suggest some people that you think might be good reviewers for your paper. And so there's this wide variety of different kinds of people who could be reviewing your paper. And there's this wide variety of people with different amounts of time to spare and different expertise and interest in reviewing your paper. And I think that the fact that we have all of that variation just left to be figured out by a single editor is just not a great way of organizing peer review. And what it means is that the average time it takes to get a publication out after you've sent it to a journal is usually several months, and in some fields it's a few years. And I think that aspect of how long it takes and also how it's organized and how it's kind of sent out to reviewers could be improved quite a lot.
Before we dive deeper into peer review, can you tell us a little bit about the history of peer review? When did it start? How did it develop? How did it become popular?
Sure. So peer review, I think the first instance of peer review was in the 17th century. And that was by the Royal Society in the UK had this journal, I now can't remember the name of, but they had, so the Royal Society was this group of scientists and so on who did lots of projects, but also had this journal that they commissioned and printed out and sent around to scientists. And there was only one person in charge of being the editor on that journal who didn't have that much contact with the rest of the Royal Society. And so he was basically in charge of deciding what to publish. And it was there was not very much of a filter in what he decided to publish. And also he obviously didn't have expertise in every topic that he decided to print. And so the quality of a lot of publications was very low. And I think after he died, there was a short period of time where they were looking for a new editor to replace him. And in the meantime, there was another scientist who wrote this satirical book chapter about this journal, giving examples of some of the terrible and stupid papers that he had published during his time there. And this meant that the Royal Society saw it as this huge blow to their reputation that they wanted to protect. And so what they did when they invited another editor to replace him was that they decided to form a council of people who would each decide which journals to publish. And I think along the next decades or so, they tried out lots of different ways to do that. So at one point, they required all of the members of the council to unanimously agree on whether a paper should be published. And that was very hard because you have to get everyone together at the same time. And also there would often be disagreements between them, and it would just take a very long time. But it took a while for them to decide to move from that version of having a council of editors to having peer review where you actually send it out to some other person who's not directly involved. And apart from the Royal Society, the other big journals that we know of today, like Science and Nature and Cell, they only instituted peer review in the 20th century. So I think for both Nature and Science, it was in the 1970s, so after World War II. And it was a similar reason in that you wanted to filter out the very low quality publications at first. And the reason was mainly to protect their reputations. So Nature is a very, very prestigious journal because they want to institute this filter and only having science papers that they think are at a high quality. And the reason that they've began this peer review process was intended to deal with that. So I think with Nature, it was specifically because they were trying to expand the range of their publication to also include scientists in America and the rest of the world. And the editor at the time talked to lots of American scientists about this and found out that they thought that Nature was kind of like this British club system where only people in London or Cambridge were being published, and the editors didn't actually care about anyone else. So they started peer review as a way to give submitted papers to people in other countries and other fields that they weren't personally aware of, in order to build up their reputation in abroad, so in America and other places.
So you mentioned how much editorial oversight there used to be prior to the broad use of peer review, and it still seems like there's a fair amount of editorial discretion. But aside from peer review and obviously the perspective of editors, what other alternatives are there? Obviously, everybody could just publish, but aside from that, what other methods are there to filter the wheat from the chaff?
Right. So I think currently the other ways that you could publish science are through preprints, and I guess even just sharing your data and code on their own on websites. So there are a lot of preprint journals now where people often use that as a stepping stone to then publishing in a print journal. But you'd have your working paper there where you can receive comments from the wider academic community before you then submit it to a journal. Or in some cases, some journals have a post-publication peer review system where they publish your paper and then they ask for reviewers to comment on the paper and then publish the reviewer comments beside it. I would say that a newer and maybe underrated form of publishing is just to put the data and code out there on GitHub or a website or something like that, where you can just look at exactly what's being done, how that results in the charts or whatever that are in the paper, that would be in the paper. And it also means that people can very easily access your data based on that. The problem with doing that is that it's completely up to each of the scientists whether they want to open that up to comments and discussion. So if you just had like your own website where you're publishing all your stuff, you might not have like a comment section, or you might not have links to where people can post reviews or responses to what you've done. And so that kind of discretion is probably not what we want. And so what I think is that I think we should just see this as like an opportunity to change how we do publishing. So the current peer review system and the current like scientific publishing system is all based on print journals that were started hundreds of years ago. And we should see the internet as like giving us the option to develop new tools to do peer review and publishing on these like platforms where you could have all of these things integrated together. You could have the data, the code, the manuscript, and reviewer comments in the same place. And I think that's probably the way to go forward.
You've brought up a number of the big problems with peer review, including things like if you publish in a journal, it's static and information changes over time, and there's not always the best incentives for people to do peer review for your work. And so it ends up taking a lot longer to get science out there. And sometimes peer review is used as a way to have the person who's doing the peer review say, hey, you have to cite my work. So it ends up sort of restricting really, truly innovative perspectives being published. I guess I'm curious. And then also there's the financial perspective of the current journals like Nature and Cell, who don't want to give up this prestige that they've accumulated by filtering out the chaff. So I guess my question is, how do you, with the internet and new possibilities for posting data available for everyone to work with, or having more interactive texts, or having more transparency when it comes to who's doing the review, or giving credit to the people who are doing the review, like how do you incentivize, how do you get those journals to go along for the ride and sacrifice their like 30% profits or whatever they're making?
Right. I mean, I think in this case, it's something where having a top-down approach is actually really effective just because of how entrenched it is. We try to publish in Nature and Science because they're prestigious, and that helps with our careers and so on, adds to our CVs. And so even if we wanted to publish more openly, there's often this disincentive to doing that. Because that stuff doesn't get highlighted in your career progression. So I think so recently I think the White House in the US released this new memo on scientific publishing where they required any publicly funded research to have their data made available, I think within the year or something. And then I think in Europe, there's often requirements on grants that are being given out where you have to make your data or code available if you're receiving these big grants. And I think that's probably the best way to do it.
Has there been any evidence that these journals actually publish better research than other journals? I know that there was something I saw at some point which showed that the number of failed replications was essentially equal between, Nature and the next tier down and the next tier down. But I'm not sure if you probably know more about this subject than I do. I'm just curious if they deserve what they've been given in terms of social status.
Right. I think there's been research on two different aspects of that. So there's the research on whether sort of high impact journals, which are the ones that have much larger readerships, if they publish better research. And then there's also looking at grant funding, which is also decided by peer review. So do decisions on who gets awarded grants do better if they have more reviewers and so on. And I think for the latter, there's some good evidence that peer review is effective at at finding researchers who have kind of both innovative and impactful research. So having more reviewers is effective up to a certain point, but also having a peer review system is much better, or not much better, but like better than having just a random allocation. And then in terms of journals filtering research, I think the evidence there is weaker, but it's also a little confusing. So I think there have been a bunch of different studies looking at this. And one example, I don't remember the name of the paper, but what they looked at was, I think, publications in chemistry where you're looking at a specific aspect of like the chemical composition of something. And I think these were for different types of molecules. And so being able to publish an accurate representation of that molecule was itself a publication. And the quality of those didn't seem to vary according to the impact factor of the journal. And so that was given as like one example of evidence where it was like the filter wasn't working. I think for other fields, it's probably harder to tell. And there's also a lot of selection into people who know that Nature is a very demanding journal will probably not submit their work there. And so there's a filter without them actually going through the peer review process to begin with. And I think there's also, you kind of expect peer reviewers might ask for certain analyses to be done or certain steps to be taken in your methods. And so that would change how you do your analysis. And I think that's kind of an important thing to remember, because if we had a system where people are just posting preprints and stuff, they don't have to go through that difficult process to get something published. They might not decide to add those like sensitivity checks or quality checks on their work beforehand, because they know that they might have this chance of being rejected in some cases, and that might be a very high chance.
How do you think that OpenAI and other language learning models will change scientific publishing and / or scientific reviewing?
So I think the current version of like ChatGPT, for example, it's probably quite useful to get like a very broad or general level understanding of a topic that you're not familiar with. One thing that I've noticed with peer review is that different reviewers have different backgrounds and expertise. So I recently had this paper that was reviewed by 6 different researchers, which is kind of unusual to have that many on a single study. But each of them pointed out quite different things. Some of them didn't really mention anything at all, and some of them just said like, this paragraph doesn't really make sense to me. Another one made very detailed comments on the specific methods that I was using. And so I think to some degree it would help reviewers understand sort of the background of a field that they're not familiar with, if that was something that was not their expertise. And so I can see it being helpful in that regard to just have this basic idea, or maybe asking questions about like, what is a common assumption made by this method? And that might give them an idea of what to ask of the researchers. Otherwise, I think it's quite difficult to tell, because I think a lot of research that's at the frontier wouldn't be captured by what's in these in these big models. So it might help you to understand methods that you're using or understand the past literature, but it wouldn't help you as much to kind of make breakthroughs. And so maybe I could see it as helping people get closer to that frontier faster, but I can't see it being that helpful at that level after that, except maybe in particular use cases. But I don't think that's like I don't think that's a use case of language models. for example.
What do you think about writing quality over time? When I read a paper from 1920 or 1950, you read Watson and Crick's one-page paper on the DNA, the structure of DNA, you read some of Einstein's papers. They're so much simpler, easier to understand, more enjoyable to read than anything that's being published today. I've thought that maybe some of this has to do with too many cooks in the kitchen. When you try to make a TV show that is one person's vision with a couple other people helping out, you form something very specific and often better than if you try to make a TV show where there are 20 different people in the room and 20 different opinions, and you kind of have to arbitrate by committee what is the appropriate phrase. In some ways, that is what AI writing sort of reminds me of, like writing by committee. Right. I'm curious how you feel like the change in that, which seems to happen around the time that peer review becomes more prominent. Do you see a connection there at all? How do we get science writing to be more interesting now?
Yeah, we recently had this really cool article published on that topic in Works in Progress called The Elements of Scientific Style, I think. I think the broad ideas that I have about that are that firstly, we have much more specialization in science now than we did in the past. So we have much more concentrated and developed fields. And so if people are only used to talking to other scientists in their field, they won't feel like they have to have the need to explain a lot of background context to their papers. And so you see this gradually rising use of jargon and abbreviations over time in scientific papers. And I think that's one of the reasons for that. I think another reason is to do with space. So often journals will have word limits, which mean that you have to put together this big project in like 3,000 or 4,000 words. But also the paper has to do loads of different methods and quality checks and so on. And it's hard to do both of those together. So you'll often have like a very long supplementary document and then a tiny paper that's just trying to condense everything that needs to be known. And then also trying to cite all the right people in the field so that because you're friends with them or like you think that they should be credited for their work. And so doing all of that together is quite difficult. And I think that probably contributes quite a lot. And then I think another area is that scientists are often not writing for a public audience in the same way as they were in the past. So if we're publishing in journals that are behind paywalls, and we're only publishing really for other people at big universities that have access to that. We're not really publishing for a wider audience. And so there's just not that like need that scientists don't have a need to write for a much wider audience. And I think that probably contributes quite a bit. It's hard to think of like what science would be like if it was written for a general audience and what it must be like. I often think in my own role, what it would be like if I didn't have access to all of these journals. And I think my whole career would be very different to what it is now. And I think that paywall kind of contributes quite a lot.
So let's say you get put in charge of everything and you get to make, one or two reasonable changes in either the process of peer review or publishing generally. What do? What are those targeted changes that you'd like to see made from the top down?
I think wherever possible, it should be a requirement for people to share their data and code. So obviously there are privacy concerns, and so that wouldn't really apply everywhere. But whenever you're using a data set that you can anonymize, it's extremely helpful to have both the data and code available, just because a lot of the specific kind of coding methods that you use might have errors in them, or they might even be broadly useful for people in other fields or in your field who want to build on your work. And so knowing exactly how you've done that can help save a lot of time, and also can help other people check whether you've made the right decisions in your analysis. That's, I think, the big one. I think the other one I would say is pre-registration. So I think this doesn't apply to all kinds of research, but pre-registration is this idea that you declare how you're going to study your data before you actually start doing that. So often you'll have the usual way of doing science is surprisingly not like that. So you'll have good data, and then you decide how to analyze the methods. You might find something significant, or you might not. And then depending on that, you might decide to change the way you're analyzing your data. And what that means is that there's a lot of room for cherry picking and a lot of room for what's known as p-hacking, which is changing your methods in order to get a significant result. And so just having these like arbitrary changes in your code or your methods might then mean that you find this significant result that means journals will be happier to accept your study to be published. And so what pre-registration does is that you tell people upfront which methods you'll be using, and you try to do that as carefully and as much detail as you can, which means that you stick by those methods when you actually look at your data, and then you don't have as much room to manipulate things afterwards if you don't find the results that you're looking for. What's difficult about that is often you don't actually know what the right method to use is, or you're new to using this data set or something like that. And I think often a way to get around that is by doing a pilot round of analysis on like a smaller data set, or like something else that you're not actually interested in, just to make sure that your code works, and then to pre-register your method. And so that tends to be, I think that's a growing practice, especially in psychology. But I think it would be a great practice for more fields to adopt.
Now we have just a couple random questions thrown in before we jump into the rapid fire. First one is, you wrote a piece not long ago in The Guardian about human challenge trials. Can you tell us a little bit about the history of challenge trials and what utility they offer over other conventional, I shouldn't say other, over more conventional methods of testing vaccines or medications?
Sure. So I guess I'll start by explaining what a human challenge trial is versus a normal trial. So that a normal clinical trial is with a vaccine is you give some people a vaccine and you give some people a placebo, and then you just sort of wait until they either get infected or they don't with some kind of pathogen. And then you look at whether the group that got the vaccine had different rates of the disease than the group that had the placebo. And so depending on how common that disease is, it could take a really long time for the trial to run. So you might have to wait several months, often several years, just to make sure enough people, even in the placebo group, got infected by the disease so that you can see whether the vaccine had a benefit. And that often takes a lot of time and cost. And it also means that a lot of people need to be involved in your study in order for you to get to find a significant result, if there is one, in the time frame that you want. So what challenge trials do instead is, with a much smaller sample, you give some people the vaccine and some people a placebo, and then you deliberately infect them all with the pathogen, and then you see if there's a difference in the rates of disease between the two groups. And so what that means is that you can kind of infect the participants in a much more controlled way, and also in a much more controlled setting. So because regular trials take place over months or years, it means that people are going home. The volunteers aren't just in the lab the whole time, whereas in a challenge trial that might be in a lab over a period of 1 or 2 weeks. And all of this whole setting is much more controlled. You can measure, you can take measurements of different samples while they're there. And you can also see whether they've developed the disease in a much shorter amount of time, because you're controlling exactly when they have that exposure. So there are a lot of caveats to doing that. The first one is that you often don't even know how to culture the pathogen in a lab in order to give them the infection. Also, the way that you're able to infect the person might not be the same way that they get infected naturally. So maybe you have like a respiratory pathogen, but you only know how to inject it into their arms. And so then knowing whether the vaccine was effective in that little challenge trial might not tell you about how effective it might be outside of it. And then there's also the dosing. So if you're deliberately infecting someone, you might give them a massive dose that they would never be exposed to in the outside world. And so then there's a question of like, how does the vaccine efficacy in this trial translate to how effective it would be outside? So keeping those limitations in mind, there have been several examples of where challenge trials have been very helpful in vaccine research. So malaria is an example. The 2 malaria vaccines, I think one was approved last year and the other is on track to be approved next year, were both tested first in challenge trials. And then various other diseases as well, like RSV, even cholera and tuberculosis had like drugs tested on them in challenge trials. One example that I like is Zika virus disease, which now is only really feasible to be tested in challenge trials because the disease is not common enough. So there's a massive outbreak around the world about 10 years ago that then kind of faded away, which means that we don't have enough people in the real world who would be exposed to it in order to test the efficacy of the drug or the vaccine. But at the same time, we expect for there to be another wave at some point. And so in order to prepare for that, the only feasible way to do that now is to design a challenge trial. And in this case, the main outcome of the thing that you would want a vaccine to do is to protect pregnant women from getting infected because Zika virus can cause childhood deformities. And so it's kind of hard to then imagine why somebody would want a challenge trial for that. You wouldn't want to infect pregnant women, obviously. But that's not what the challenge trial would involve. Instead, it would involve women of that age who are given contraceptives and are not intending to become pregnant during the trial. And then what you would do is test whether they were able to completely prevent an infection by taking blood and urine samples and making sure that there wasn't any virus detected in those samples. So you would then be hopefully confident that if they were able to completely block an infection, then they wouldn't be able to pass that on to the fetus. So I think that's now being started for Zika virus vaccines, but I can also see how it might be useful for others that are neglected at the moment, just because it's too difficult to enroll enough people in the sample, or maybe the disease is not common enough, but we expect a future wave.
So a few years ago, you were on the podcast Rationally Speaking and discussed gender differences in brains, brain differences between the genders. Have you changed? How has your thinking evolved since then at all? And what are your current thoughts on intrinsic differences between the genders in terms of their brains?
Yeah, it's been so long since I did that podcast. I can't remember exactly what I said. I think some of my friends listened to it and they were like, I don't actually know what your position is on this topic even now. And I think my conclusion there was just that the research that's being done in that area is not actually very good at distinguishing between innate and environmental differences between men and women. So often what they'll do is they'll look at they'll compare children's brains at a very young age, and then they'll also compare children's brains at a much older age and say, well, there weren't these differences in infancy, but there were when they were 10 or 11 years old. And that must be the results of environmental factors or cultural factors. And I think that is a reasoning process that doesn't really make very much sense, just because a lot of genetic influences take time to show up and develop. And so we know that from, I think the most obvious example is like Alzheimer's disease, where you have this genetic variant that increases your risk of Alzheimer's disease, but you're only going to notice the effects of that when you're very old. And so you have these changes that are kind of expected to happen at a much later date than you would expect to see in infancy. And then I think there's also that question, not just looking at brains specifically, but also just differences in behavior, differences in performance on various like subjects, so maths or English or whatever. And if you're comparing children at a young age and saying, well, there weren't any differences between boys and girls at that time, but there were a few years later, and that means that it's environmental, I think that also doesn't really make sense for the same reason. But at the same time, there is lots of evidence that like role models and gender discrimination can affect people's performance. And so I don't think it really has to be either or. I think that they can both contribute. And probably each side of this debate is underestimating the other side. And it's just a question of how we're studying this that I think is not usually done very well.
All right. Well, we have a couple of rapid fire questions for you. Mm-hmm. What important medical truth do very few of your colleagues in your specific field agree with you on?
I'm actually not sure. I don't know what contrarian opinions I have in that area. I think most people probably underrate like the value of medicine and healthcare in general. People don't realize how much of an impact it's had on mortality and on people's lives. I think one reason for that is that we often only think about healthcare when we need to go to the hospital or when we develop condition. So we think, okay, well, this drug isn't working for me, and therefore medicine is not working in general. But we don't think about all of the conditions that we haven't even developed at all because of medicines that we've had at a much younger age, or because of vaccinations and so on. And so I think people really underrate the amount of progress that we've had in medicine. That's probably the biggest thing.
What is one scientific article that you think more physicians should be familiar with?
I would say maybe the same sort of answer. I think that more physicians should be aware of how much has changed in terms of healthcare and people's outcomes. So on Our World in Data, we have a lot of data, a lot of charts and metrics on like childhood mortality and maternal mortality and so on, and how much these have changed over time, how much life expectancy has increased. I think the thing that often people don't realize is how much life expectancy has increased also at older ages. So people often think that most of the progress has only happened in terms of reducing mortality at childbirth. And then as long as you live until 5, we aren't living much longer than people were in the past. And that's not true. You actually see gains in life expectancy throughout the lifespan, and they're also very big. And so I think that's something that more people should be aware of.
If you had unlimited funds for a single randomized controlled trial, you get to investigate anything that you're interested in, what are you going to fund?
Probably more vaccines for neglected diseases. So one thing that's very difficult with, like even with malaria vaccines, which I think have been a goal for several decades, is that getting the funding for them is very difficult because it's not something that affects a lot of the countries with that funding. So like the US hasn't had malaria for many decades, and it's also just not a public health concern in those places. So I think just being able to fund the research that already could happen to a much larger scale would be a big improvement.
And the last question is, what should we have asked you in this interview, or something that you really wanted to talk about, but we didn't actually talk about?
I kind of just go with the flow and like whatever you guys are interested in talking about. A thing that I was working on recently that I think isn't that well known is the ongoing eradication program of guinea worm disease. So I don't know how much you know about that, but basically guinea worm disease was this very debilitating disease that was quite common in a lot of African countries and South Asian countries in the 1960s and before that as well. And starting from the 1970s, a bunch of big public health groups, including the Carter Institute, the WHO, and UNICEF, all got together to try to eradicate the worm. And doing that was surprisingly easy. I think it's often called like the easiest disease to eradicate because the way that it is transmitted is these worms contain larvae that cause so the worms release larvae into the water, and their larvae can be taken up by small water fleas, and those water fleas are then consumed by people. And eventually the larvae invade their intestines and develop into this very long worm over several months, and eventually form a blister on some part of your skin and erupts from that. And because that's so painful to people, they often then go into the water to relieve the pain, and then that releases more larvae into the water, which restarts that cycle. And that process takes about a year. So there's like around a year usually between when someone's infected and they develop the blister that releases more larvae. And so there are two things that make it surprisingly easy to eradicate. One is that the time span that it takes. to release that infection. And the second is that the water fleas are not able to survive very long outside, or the larvae aren't able to survive very long outside of humans. So if you're able to filter drinking water, or you chlorinate the water, or even if there's like a drought in some year and all the water fleas die out, you could completely eliminate the disease in a particular village. And so what this eradication program involved was just lots of water filters, public education about where to drink water safely from, how to identify cases of guinea worm disease and prevent them from then going into the water and releasing further larvae. And it's been incredibly effective. And so whereas there were around 1 million cases, I think, in the 1970s per year, there were only 15 last year. And so it's most likely going to be eradicated very soon. And it's a huge success story.
Nice.
Public health.
Excellent. Excellent. Saloni, thank you so much for joining us on the External Medicine podcast. Before we close, could you tell people where on the internet they should go to learn more about you?
Sure. You can find me on Twitter at Salonium, so SALONIUM. I also have a Substack newsletter where I need to restart that, but it's a weekly newsletter where I keep people updated with the latest scientific research that I find very cool and interesting. And I try to keep that limited to stuff that I know about and that I think is high quality. And then you can also find me at Our World in Data, a website that presents data and research on the world's biggest problems.